Hello everyone, my name is Harika Ürel and I'm PhD student in Lara Urban's lab at Helmholtz Munich. Currently, I'm working on Squiggle analysis for metagenomic viability inference. So metagenomics allows us to study whole microbial communities found in environmental samples such as water, soil, and air. One of the disadvantages in metagenomics is we cannot differentiate living and dead microorganisms. We use nanopore sequencing to test the hypothesis if Squiggles can detect differences, what we call viable and dead DNA. Since Squiggles can detect changes down to the atomic level, such as epigenetic modifications, we would like to build a computational framework that will combine nanopore sequencing and neural networks to distinguish living, therefore pathogenic microorganisms from dead microorganisms. And of course, to train a model, first we needed to generate the data, and we achieved this by culturing E. coli for 42 days and induced cell death by not providing any nutrients starting from day 3. We separated DNA from living and dead microorganisms via centrifugation, where we find this dead, free-floating DNA in the supernatant and viable DNA in cells in the sediment. Later, we used nanopore sequencing to generate squiggles from these DNA fragments. And as we expected, we observed an increase in DNA concentration of the supernatant and decrease in the sediment. We chose the same model architecture that was employed for a similar Squiggle application, which worked really well. Here which is the one-dimensional residual network. Here we have 4 residual blocks, and each contains 2 bottleneck layers with convolutional layers, batch normalization, and activation functions, followed by skip connections. And the final layer is to classify if the DNA is dead or alive. As a result, with the area under the receiver operating characteristics curve of 0.7, we achieved a reasonable trade-off between recall, which is the sensitivity, and the specificity. And the area under the precision recall curve reaches to 0.65. Here we have the overview plot for all of the metrics that we have. And as we can see, the highest we achieved was the recall with 0.77. However, the model detects viable DNA well. However, the specificity is pretty low. And we think that this might be caused by a mixture of dead DNA in the supernatant, which died in different time points. So currently, we are working on for hyperparameter optimization and we would like to apply explainable AI techniques to see which features are recognized by model to make classification. And we are working, exploring this alternative experimental methods to avoid this mixture of dead DNA. So I would like to thank to my group and to the collaborators, and thank you for listening.